So next bacteria we are going to discuss is the Haemophilus. So the genus Haemophilus, it contains non-motile, non-sporing and gram-negative bacilli. So the genus Haemophilus, it contains the small non-motile, non-sporing, oxidase positive, gram-negative bacilli. The Haemophilus word, it means the blood loving. So they are characterized by the requirement of one or the two accessory growth factor like growth factor number 10 and growth factor number 5 present in the blood. That is why it is known as Haemophilus, because they are blood loving. So the different species have the different requirement. For example, the Haemophilus influenzae, Haemophilus aegyptius and Haemophilus hemolyticus, they require the factor number 10 and factor number 5 for their growth, while the Parainfluenzae, Parahemolyticus, Paraphophilus, it requires only factor number 5 for their growth. The Haemophilus ducreyi and the Haemophilus Apherophilus, they require only factor number 10 for their growth. Right? Now starting with the first Haemophilus, that is the Haemophilus influenzae. So the Haemophilus influenzae, these are the small with the size of 1.5 micrometer into 0.3 micrometer, and these are gram-negative non-motile bacillus, which shows the considerable polymorphism. Important point is these are non-sporing and non-acid fast. Right? So the morphology, they are gram-negative, cocobacillary capsulated bacilli. Right? So the culture, it requires the factor number 10 and the factor number 5 for their growth. They does not grow on the blood agar. Very important. The influenzae, they does not grows on the blood agar. The media used are the chocolate agar, the Leventhal medium and the Fields agar, which is best for the primary isolation. Important. Right? So in this there are 8 biotypes that have been identified on the basis of indole production. Indole production, urease and ornithine decarboxylase activity. So biotype 1, it is most frequently responsible for meningitis. Important. So in H. influenzae, the biotype 1, it is most frequently responsible for the meningitis. So the major antigenic determinant of the capsulated strain is the CP, which is the capsular polysaccharide, which is based on the H. influenzae. Strains have been classified into 6 capsular types from A to F. So type B, it is responsible for 95% of the cases of the influenza meningitis. Important. The strains that lacking a capsule cannot be typed, and these are called as non-typable strains, in which the strains lack the capsule. These are known as the non-typable strains. The capsule protects the H. influenzae from the phagocytes. Important. So what is the role of the capsule? It basically protects the influenzae from the phagocytes. Coming on to the lab diagnosis. So in this, the most reliable method for establishing a diagnosis of HIV infection is the recovery of the organism in the culture. Important. So the detection of the capsular polyribosyl ribitol phosphate, which is the PRP, which is the polyribosyl ribitol phosphate. It is an important adjunct to culture in the rapid diagnosis. So the method used are the immunoelectrophoresis, the latex agglutination, coagulation and ELISA. Right, so these are the method. So the treatment is the cefotaxime and the ceftriaxone. These are the two drugs of choice. So administration of the glucocorticoids to the patient with the HIV meningitis, it reduces the incidence of the neurological sequelae. So the vaccination is the conjugate vaccine. It is available for the H influenza type B. HIB, it stands for Haemophilus influenzae type B. So currently no vaccine is available for non-typeable H influenzae. Discussing the pathogenicity of the influenza. So it does not secrete any exotoxin. The polysaccharide capsule, which is the non-polypeptide, it is considered to be the major factor in virulence. Important. So there are 6 types of the capsulated designated as A to F. Of these, type B, it is commonly associated with the invasive influenza disease in the children. Important. Type B. So the polysaccharide capsule of type B strain, it is composed of the PRP capsule and it is proven determinants of the virulence. So the PRP capsule, it exerts its effect in the following ways. Number 1, it is anti-phagocytic. It does not induce the alternative complement pathway. So for this reason, the anti-capsular antibody, which promotes both the phagocytosis and the lysis of the bacteria, it is the main factor in the immune defense against the influenza infection. Important. The capsulated strains cause the invasive disease, among which the meningitis is the most common. Right? Coming on to the host defenses against the H influenza. So it is susceptible to lysis by the antibody and the complement. Therefore, the host defense against the influenza include the activation of the alternate and the classical complement pathway and the antibodies to PRP capsule. Important is the antibodies to the HIP capsule, Haemophilus influenza type B capsule, play the primary role in conferring the immunity against the H influenza. These bactericidal antibodies directed against the PRP capsule of the H influenza type B, it is directly responsible, or it is entirely responsible for the host resistance. So in the newborns and in the infants, these are protected from the infection up to the age of 6 months. Important. Why? Due to the placental transfer of the antibodies against the B capsule from mother to fetus. Right? So when these transplacental antibodies to the capsule wane, infants, these are at a higher risk of developing invasive H influenza disease. Important for your MCQ point of view. Now coming on to the infection which are caused by the H influenza. So it is considered, or it is divided into 2 types, the invasive diseases and the non-invasive diseases. In the invasive diseases, it is caused by the capsulated strain of group B, which accounts 95% of the cases. Important. The bacillus acts as a primary pathogen causing the acute invasive infection, and the bacillus spread through blood. Spread through blood, and it is typically seen in the children. The types of infections are the meningitis, which is the most common. So if the question comes, which is the most common type of infection which is caused by the H influenza, so your answer should be meningitis. It is the most common. Brain abscess, acute epiglottis, laryngoepiglottis, bacteremia, abscess, pneumonia, arthritis, endocarditis and pericarditis. So these all are the invasive diseases. Coming on to the non-invasive diseases, so these are caused by the non-titratable strain. The bacillus spread by the local invasion along the mucosal surfaces and cause the secondary and the suprahepatic infection. The invasive diseases, these are typically seen in the children, while the non-invasive diseases, these are seen in the adults. So the type of infections are the otitis media and the sinusitis. Now let's discuss the important point which is related to the Haemophilus influenzae. So this is the satellitism. So what is satellitism? It is the growth of the Haemophilus influenza in a scanty, first of all, the growth on a blood agar. As a factor 5 is not freely available, being imprisoned inside the red blood cell or the RBCs. So in this the growth therefore better if the source of factor 5 is also provided. So when the Staphylococcus aureus is streaked across the plates, this is the streak of the Staphylococcus aureus across a plate of a blood agar on which the specimen contains the influenza has been inoculated. So after the overnight incubation, the colonies, these are the colonies of the H influenza, these will be large and well developed along the side of the streak of the Staphylococcus and smaller farther away. So this phenomena is known as satellitism. So in this is a streak of a, this is a streak of a Staphylococcus aureus, and in this the colonies of the H influenzae, these are larger and these are well developed along the side of the streak of the Staphylococcus, while these are smaller which are farther away from the streak of the Staphylococcus aureus. So this is the streak of the Staphylococcus aureus. The colony of the H influenza, these are larger and these are well defined or well developed along the side of the streak of the Staphylococcus aureus, and these are smaller when they are farther from this streak. Fine, that is why it is known as the satellitism. So the satellitism it is due to the high concentration of factor number 5 in the Staphylococcus aureus, which is released into the medium and it is used by the H influenza. So in this the satellitism is due to the high concentration of the factor number 5 which is released from the Staphylococcus aureus into the medium, fine, and it is used by the H influenza. So this is all about the Haemophilus influenzae. Now second is the Haemophilus ducreyi. So these are the gram-negative, but it may appear as a gram-positive, and frequently these shows a bipolar staining, but mainly these are short gram-negative Coccobacillus, which is ranging from the size of 1 to 1.5 micrometer to 0.6 micrometer, and in this they have a tendency to occur, important point, in groups or in parallel chains. And they usually take the bipolar staining. Fine. So in this, the microscopic finding in this, it shows or it resembles a school of fish, a school of fish appearance. So it is a gram-negative but often may appear as gram-positive, frequently shows the bipolar staining. The bacilli may arrange in the small groups or in a parallel chain, which gives us school of fish or a railroad track appearance. Fine. So chocolate agar enriched with the isovital X or the fetal calf serum and containing the vancomycin, it acts as a selective media. So these are the selective media. So it causes the chancroid or the soft sore. So these are the painful, non-indurated, irregular ulcers. These are the ulcers which bleeds easily, and these are enlarged tendal inguinal lymph node. Important. So the diagnosis, acute diagnosis of the chancroid relies on the culture of the Haemophilus ducreyi from the lesion. So the treatment is the single dose of the azithromycin is the drug of choice, while the erythromycin, these are the alternates. So these are the two important points. Please don't get confused between the chancre of your primary syphilis and chancre of your H. ducreyi. See, this is the hard chancre, which is the ulcer of the primary syphilis, while the soft chancre, it is a chancroid, it is the ulcer caused by the H. ducreyi. So this is the hard chancre in the primary syphilis, while the ulcer caused by the H. ducreyi, it is soft. And don't get confused in between these two. So guys, this is all about the Haemophilus. Please go through it once and try to make the notes. Thank you.